Good afternoon, everyone, and welcome to the second PSI webinar from the Special Interest Group in Toxicology. As some of you may know, last time we had a very interesting presentation by David Lovell on the AIMS test, but today it's my honor to introduce to you Professor Dr. Thomas Yaki. Thomas Yaki is Professor of Statistics at Lancaster University and Director of the Medical and Pharmaceutical Research Unit, which has a long-lasting tradition in the design and analysis of clinical trials. He's an NIHR Senior Research Fellow, coordinator of the famous EU-funded IDEAS network, and a co-investigator of the MRC's Northwest Hub for Trials Methodology Research. His research today has focused mainly on adaptive designs, Bayesian methods, and preclinical statistics. He has worked on estimators for pharmacokinetic parameters, developed adaptive designs, in particular for multi-arm studies, and investigated Bayesian methods for dose escalations. And today he will present for us on optimal designs for non-compartmental analysis of pharmacokinetic parameters. Thomas, I give the floor to you.
Thank you very much for that very kind introduction, Helena. And I must say it's probably the first time that anybody has called the IDEAS network world famous, so thanks for that. I'll be talking about optimal designs and optimal designs in the context of non-compartmental analysis of PK parameters. Just to start off with, I'd like to point out that really all the hard work behind this presentation has been done by Helen Barnett, who's just completed PhD student that worked with us on that project. And we're also very grateful for the input that Tom Jacobs and Helena had onto this work. So what are PK studies? I'm sure this audience doesn't need to be reminded of what they are, but I'll do it anyway for completeness sake. Pharmacokinetic studies study what the body does to a drug, and frequently it measures the concentration of the drug in the blood. Common measures, common PK parameters of interest are, for example, the area under the concentration curve as a measure of drug exposure, or Cmax as the maximum concentration. Now, typically, a PK study is conducted in the following way. There is a certain number of subject animals in our context here that are sampled, blood sampled, plasma sampled at a given pre-specified set of time points, and then the measured concentrations are plotted against the time as shown here in my illustration. And from these data, we then try to derive relevant summaries of that black curve that you see underlying here. So the challenge really in this PK study context is that, well, we obviously don't know what that black line is. And so to some extent, we want to understand what the relationship is between the concentration over time. And at the same time, we want to have meaningful summaries, meaningful measures for this behavior of changing concentration as time goes on. Now, if you look at that illustration, you can see that I have different colored dots in my graph, and this is not just to pretty up my graph. These are supposed to represent different animals that have been sampled. So, for example, you can see that there is a set of red dots that are in the first half of the plot, one at around 1 hour, one at 2 hours, and one at 4 hours. And so here we're thinking that this one animal has been sampled at 1 hour, 2 hours, and 4 hours. And that is going to be useful to give us some understanding about the correlation between the measurements at the different time points. And at this point, the question for us has become, okay, so now that I have a certain set of animals that I can study in my study, and I can take, say, up to 3 different measurements per animal, how should I distribute when I'm going to sample my animals, when I'm going to take my blood samples? Recently, in 2014, Chapman et al. Proposed study design for toxicokinetic analyses for GLP rat studies. And this is what the proposal looks like. So in essence, what they are saying is the first animal should be sampled at time point 1, 4, and 5, whatever that corresponds to in my previous graph. So, animal 2 gets also sampled at time point 1 and 4, but rather than being sampled at time point 5, it's going to be sampled at time point 6, and so on and so forth. Now, clearly, if you look very carefully at that scheme, you notice that it has a couple of balancing properties, and I guess if you look closely, it seems like a reasonable choice to sample to use in such a PK study. For us, the question then became, so, okay, well, this is certainly a reasonable design to use, but what is the best possible design? And so our objective of today, or the objective of this work, was to investigate whether we can find an optimal sparse sampling scheme. In other words, identify how we can optimally choose when to sample which animal. Now, for those of you familiar with the area, you'd think, well, that's a solution that's been given a long time ago. Well, there's two additional constraints that we're going to put on our objective. The first one is we want the design to be optimal for non-compartmental analyses. So we're going to make use of non-compartmental methods as much as possible, and I'll highlight on the next slide what I mean by that. The second requirement is that we want the design to be robust, because one of the fundamental thoughts behind the non-compartmental analyses is that we don't need to make many assumptions about our data generating process. Now, you'll see that unfortunately we can't completely go away from assuming something, and therefore we still want to make sure that in the spirit of non-compartmental analyses we obtain robust optimal designs. Okay, so if we just focus for a second on the area under the concentration curve, and let's pretend we are in the perfect situation that we know what that underlying time to concentration relationship looks like. In other words, let's assume we know what that underlying black line is going to look like. Then the area under the concentration curve is simply the integral from 0 to the last observed time point over that black line, that ytk in my notation here. Now, if we want to use a non-compartmental estimator, what we're doing is saying, well, we don't know what happens between our time points T1 and T2, and what happens between time point T2 and T3, and therefore we're not going to assume that there is an underlying model. Instead, we're simply going to linearly interpolate between those time points. And that results then in this red, well, no longer smooth curve underlying. And then on the basis of this underlying model, if you want, we can then obtain an estimator of the area under the concentration curve, which is essentially just the sum of these blue rectangles corresponding to the midpoints of each of those intervals coming from the linear interpolation. So the fundamental difference between, I guess, a more traditional or more straightforward modeling approach is that we assume we don't know what happens between points, and therefore our best guess is that concentrations change linearly in between. Okay, so with that in mind, we're going to have to think about, okay, so how can we then obtain an optimal design? Well, would we know what this underlying curve would look like, then the problem is, well, not easy, but has been solved. So there are optimal design solutions for nonlinear mixed effects models of the shape shown in this graph. But because we have this discontinuity in our non-compartmental estimator, the question becomes a little bit more tricky. And so for illustration purposes, let's start off with our first assumption that we're going to use in our search for an optimal design. We're going to start by assuming a model, and we're going to assume that it does have a sort of non-linear form. And in particular, for the example that I'm going to use, we're going to assume that we have a one-compartmental first-order kinetic model with oral administration, and you can see the non-linear form here. The values f and d are fixed constants, and the three parameters v, ka, ke are going to have random effects in our assumed true model, while the errors are going to be normally distributed with constant coefficient of variation. Okay, so how can we then obtain optimal design? Well, what we're going to do is we're going to make use of a two-stage procedure. The first stage, we are asking ourselves the question, okay, what are the best time points that we should consider in the first place? So we're going to be rather pragmatic here and say, well, we know that yes, in theory, we could sample at any possible continuous time, but in reality, there's going to be some limitations on what we can and cannot do. And among this set of possible time points, we want to find which one are the time points that give us most information about the underlying PK relationship. And then, in the second stage, we're going to assume that we have chosen the time points, hopefully according to the steps to the first stage. And then we're going to ask ourselves this question of, okay, given the time points, how can we choose what the best sparse sampling scheme is? It's probably worth saying that all the ideas that I'm outlining in the next couple of slides don't implicitly require you to use a two-stage procedure. In other words, you can conceive a similar sort of approach using a single-stage procedure. The problem is going to be computational. The procedures that I'm going to introducing can be computationally quite demanding, and therefore we've split it up into two stages rather than doing a global optimization in one step. Okay, so here is, in a nutshell, the algorithm, the procedure to choose the time points. So first of all, we start off by defining a set of possible time points. So at this step, we're just going to think about, well, it's not going to be possible to take a sample after 2 seconds post-administration, 3 seconds post-administration. We know that there are some practical limitations. Let's define what these practical limitations are. So, for example, you can't take more than one sample within a half-hour window or something like that. Then, here's the step where, and our second step is where our assumption comes into play. So we are going to assume that our model, our one-compartmental model that I've shown you before, is in fact true. And so we're going to generate a large number of complete data sets from this underlying model. And by complete data sets, I mean we generate data at all possible time points. Then we rank the time points based on how far are they from the truth. And then in our fourth step, we're introducing robustness. We're going to repeat that for these steps 2 and 3 for a couple of different candidate models. Keep in mind, we're thinking here, well, we might have a rough idea of what the model shape looks like, but we obviously do not know what it is, and therefore, to introduce robustness, we're going to evaluate the optimal time points across a set of different models. And then we use a minimax criterion, that is, we use the smallest of the worst of the ranks in order to find a robust optimal time point choice. So more specifically, here in this illustration, you can see in black, this is my assumed underlying non-compartmental time concentration curve, while in red you can see the possible deviations if I use 6 time points past 0. And so what you do is we literally take possible sets of time points that we're interested in. So if we are interested in choosing 6 time points in our sample out of the, I think it's 24 in this case, that we're allowing, we simply measure how far away is the red curve from the red observation. Now keep in mind, red comes from simulated data. And now you could think, well, actually, if I assume what the true value is, I could as well use the true value instead rather than simulating data. And that's perfectly true. The reason why we don't use the actual true value is because it allows us to incorporate uncertainty in our estimate in our underlying concentration time profile that otherwise would not be incorporated. Okay, then we're going to find for a set of grid points G for our ICE simulated data set. We compute how far away we are. We take the average over all our grid points across all our different models, and we choose the best time. Sorry, the best time point is the one that minimizes this objective function here. Okay, so then a variant to the theme, if you want, then thinks about, okay, in reality, it doesn't matter if my optimal design says I should only sample at time points 12, 13, or 14, and 14 hours. Nobody would use such a sampling design, right? Typically, you would have some predefined regions that you want some information about the concentration time relationship. And so a variant to the theme is by introducing different zones that we allow the time points to be chosen from. So for example, I could say my first zone is I need to choose at least one time point from half an hour. In other words, my first zone determines that I need to use the first time half an hour as my first sampling time point. The second zone then says, well, I suspect that somewhere between 1 and 4 hours the maximum concentration occurs, and so I want to learn something about the curvature, and therefore I want to choose 3 time points out of the grid from 1 to 4 in half an hour intervals. I want some information about the tail behavior, so I want one sampling time point between 4.5 hours up to 11.5 hours, and I force the last time point to be sampled as well. So in a nutshell, this is what we are doing. We're saying we're forcing a sample to be taken at the first time point, that's the black dot. We want 3 observations from this red colored region, and we want 1 observation from the blue colored region. Okay, so now that I can find what the optimal time points are for a given model, and I can incorporate practical constraints such as the zones that I've just introduced before, I want to think about how can I incorporate the fact that there are potentially different scenarios, different models, different truths that I'm assuming. So if I define R as my rank in my scenario P, then a robust choice of optimal design is the design for which the worst of the ranks for a given time, for a given set of time points is minimized. In other words, I'll choose the worst amongst the best. Sorry, the best amongst the worst. There you go. So for example, here is how this would turn out in practice. So here I'm considering 8 different scenarios, and the scenarios in this illustration are all drawn from the same model that I've illustrated before, but what's changing between the different scenarios is the variability of the 3 random effects. So LLL, my first scenario, corresponds to a setting where all 3 random effects have a relatively low variability. LLH means the first 2 have low variability and the third one has high variability, and so on and so forth. So I'm assuming an overall model, but I'm considering different potential variability scenarios. Then for a set of time point choices, and the numbers 225, 260, 235 on top just simply corresponds to a given set of sampling time points. I'll find what is the rank in terms of deviation from the truth for each of those choices. And you'll see that, say, for time point choices 2, 2 to 5, sorry, these numbers don't actually have any direct interpretation, but this is just my placeholder for, this is the 225th combination of time points that I could be interested in. You can see that for the low variability scenario, this is the 21st best design that you could use. For the LLH scenario, so low variability, high variability, it's the 13th best choice, and so on and so forth. And you can see that across the 8 scenarios that I'm considering, it is consistently in the region of around a rank of 30. And so the minimax, so the worst of these ranks, is 40 in this case, and hence it is the best of the 5 choices that I show you here on the screen in terms of the worst performance across the 8 scenarios that I'm considering. Notice that there is a tie, so time point choice 260, for example, also gives me a maximum rank of 40, but because the total rank, or if you want the average rank of 225 is better, we opt to go with 225 as our optimal choice of time points. So in practice, how does it look like? It means we're sampling at time point half an hour. That's the one that we pre-specified in the first place. Then remember, we had 3 choices between 1 hour and 4 hours, and the optimal time point to choose is 1 hour, as well as 3.5 and 4 hours. And then we had 1 time point between 4.5 hours and 11.5 hours, and the optimal one to choose in this case is at 7.5 hours, and then we have a fixed time point at 12 hours as the final time point to sample from. Okay, so now let's go on to the second stage of the procedure. So in the first stage, as I said, the purpose was to identify the time points that should be considered for sampling. In the second stage, we're asking ourselves the questions, given a prefixed number of animals and a prefixed number of times we can sample the animals, how shall we decide when to sample which animal? The first step in the procedure or the algorithm is we define a set of feasible sparse schemes. Basically, the first step is saying, from practical considerations, what possible scenarios do I allow? And I'll give you an example of what that will look like on the next slide. And then we're going to do the same as before. We're going to generate a large number of complete data sets from our underlying model. We rank the different sampling schemes. So for each of the schemes that we've deemed are feasible in step one, we find the rank for a given optimality criterion. We introduce robustness by repeating 2 and 3 for different models, and then we apply our minimax criteria to the ranks in order to find an optimal sparse sampling scheme. So first step is defining which schemes are feasible. So for our example, we define feasible by saying we're going to have 10 subjects and we're going to use 6 time points to sample from. And the 6 time points are going to be the 6 time points that we've deemed to be optimal from the first stage of the procedure. We're going to say that each sample can each subject, each animal can only be sampled at 3 times. So at 3 time points. In addition, we want 5 out of 10 subjects to be sampled at each time point. And we're going to make the additional assumption that schemes cannot be repeated. So if you look at the scheme for animal 1, that's going to be different from the scheme for animal 2, and so on and so forth. Now, obviously, your choice of what a feasible scheme is will depend on your context, will depend on your question. We have simply modeled our feasible schemes on the basis of the proposal that was given in Chapman, where they had 5 subjects, 5 animals per time point, and 3 samples per time point. If you wanted, you could also include further additional restrictions. So you could say, well, I don't want animals to be sampled at consecutive time points. And that could be incorporated. The trick or the difficulty is you need to be able to define every single scheme that is feasible a priori to exploring what the optimal design looks like. So under the constraints that we've put on, there are just over 1,000 different possible sampling schemes. And again, if we relax some of the assumptions, so for example, if we all of a sudden decide, yes, schemes could be repeated between animals, the number of possible feasible schemes is going to increase drastically, and hence the computational burden of the procedure increases rather quickly, as I've alluded to earlier. Okay, then I need to choose an optimality criterion. For example, my optimality criteria could be the variance of a PK parameter that I'm interested in. So that could be the variance of the AUC, or it could be the variance of Cmax, and so on. In the spirit of robustness, however, I might also consider a robust version where I have a weighted sum of different criteria. So, for example, I could consider the optimality criterion to be a weighted sum of the variance of the AUC and the variance of Cmax. If you do that, you'll find that actually the choice of criterion does matter, and it does matter notably. So the graph that you see here is for the same model, for the same data, simulated data that is, we find for the just over 1,000 different schemes, we find what is the rank of that scheme if I use the variance of the AUC, as well as if I used the variance of Cmax. And you can see that there is a bit of a clustering towards the tails, so closer to 0, 0 and closer to 1,000 and 1,000. You can see that there are sort of similar values, but in between there's a large discrepancy between which ones of the designs are optimal based on your criterion that you choose. In practice, that means simply you need to think about which one is more important for you if you have to choose one of them, or go the robust way and incorporate both of them in your optimality criterion. Okay, so now if I then apply this procedure that I've described above, using the optimal time points derived from step 1, I can do the same thing as I've done before for each of my just over 1,000 different schemes for my 8 different variants of the same model. And what we find is that, for example, the scheme that's entitled 811, and again, I'm afraid there's no direct interpretation of that number other than this is the 811th possible scheme that we've considered, we find that for all 8 of these different scenarios, the rank of that particular scheme is never worse than 10. 810, on the other hand, has a little bit more variability in the ranks. The worst rank is somewhere around 20, while once you go up to schemes 1,001, for example, yes, you could do very well in some scenarios, and if I remember correctly, the ones where the rank of 1,001 is pretty good are the scenarios where the variability is low in one of the parameters. But if the variability is large in this parameter, you get much, much worse performance. Your rank goes up to 60 to 80. Now, it's probably also worth pointing out at this stage that actually a rank of 80 isn't actually horrible in this case, considering that there are 1,000 some different schemes that are possible in the first place. So it's still a very well performing, but it's just not as good as the scheme 811. Okay, so in practice, what does that look like? What does that correspond to in terms of comparing to the proposal by Chapman? So here, on this table, you can see what the optimal sparse sampling scheme looks like. When you see an X, that same sampling scheme is proposed as part of the scheme in Chapman. Where you see a circle, it has been replaced with a different sampling scheme than the one that was proposed in the publication. So you can see that while there are 6 out of the 10 different schemes that are the same, there are also 4 that are different. Okay, so at this point, you might ask yourself, so why bother? It seems like there's a lot of work going in order to find what the optimal sampling scheme is, when I could just have used the proposal by Chapman, which, as I said in the beginning, is looking perfectly reasonable. The point is that the variability in the estimates can be reduced up to 15%. So if you had chosen the worst performing scheme out of the 1,000 and some, then your variability would have increased by 15% in comparison to the best performing scheme. Now, obviously, the worst performing scheme might have all sorts of different flaws that in practice you would never consider, but even against the reasonable scheme by Chapman, you gain somewhere between 3 and 5%, depending on which truth you're looking at. And so, given the moderate amount of effort to find an optimal design, you can actually gain a notable amount in precision of your estimates. Moreover, if I, on top of just optimizing the scheme that I'm going to use, I'm also optimizing the time points, I get a further reduction in variability of about 2.5%, so that if I use this two-stage procedure that I've proposed, I can gain up to 7.5% in variability in comparison to the proposed design by Chapman. Your other concern might be, okay, so what if I had just simply used an optimal design for nonlinear mixed effects model and used that instead? Well, it turns out that the optimal design that's optimal for the nonlinear mixed effects model is actually often quite different from the optimal design that this procedure that I've talked about gives. And so, again, comparisons are a little bit difficult because it measures slightly different things, your NCA model in comparison to your nonlinear mixed effects model. But if you compare the corresponding estimators, so had you used the optimal design from the NLME model for an NCA analysis, you would do a couple of percent worse than had you used the optimal design for the NCA directly. So what I've talked about is two different algorithms that are hopefully useful to find optimal designs for PK analyses using non-compartmental methods. What I haven't shown is that you can actually mathematically show that both algorithms will converge to the true optimal design as long as you consider a large enough number of resamples. It can be computationally expensive to do, so depending on the number of time points that you're allowing, on the number of schemes that you're allowing, it could take several hours or possibly days if you have a very large number of schemes to find the optimal design. And we are currently working on an R package called PK Design, which is implementing the methodology that I've just talked about, and hopefully that will be available soon. Thank you very much.
Thank you, Thomas, for this very interesting presentation. Now, for the remainder of this session, I would like to open the floor for questions. I invite everyone from the attendees to post questions. In order to have this organized in a kind of orderly fashion, we would like you to post your questions through the chat function, and then your questions will be read to Thomas. In the meanwhile, while everyone is thinking about potential questions or remarks to discuss, I have two discussants, Adriaan Blommaert from Open Analytics and Fitine Tekle, who I want to give the floor for the first set of questions. Adriaan, please go ahead.
Hello, thank you for a very clear presentation. I was wondering how this proposed method for finding optimal designs relate to other optimal design concepts such as the optimality?
That's a very good question. So one of the fundamental ideas behind the optimality is that you optimize your variance covariance matrix of your parameters characterizing the underlying model. Now, in the NCA context, we don't have as such an underlying model with multiple parameters. We have a single parameter at each time point, that is the average concentration at that time, and therefore, conceptually, we don't have a variance covariance matrix per se that we can build on, and hence there is no obvious relationship between the traditional optimality criteria that we're familiar with in the context of nonlinear mixed effects models, or any model, in fact, and what we are proposing here.
Okay, thank you. Another question from a practical point of view. You attach some weight to estimating the variance of the area under the curve and to Cmax. How in practice would you come up with weights? Or
that's a very good question. So the question is around here in my robust version, how do I find W1 in essence? So what we have chosen in our illustration is we weighted both components equally, and rather than looking at the variance of AUC and the variance of Cmax, we've actually looked at the scaled variance of both of them. Now, that difference might not be immediately obvious why we've chosen to work on the scaled version, but basically the idea is we're going to bring the variability of AUC and the variability of Cmax onto the same level, and then weight them equally. Because if we wouldn't do that, I think it is Cmax that tends to have a much larger variability in the estimate, and therefore, even though my weighting would be equal, I would get more contribution from optimizing with respect to Cmax. So we first scaled and then applied an equal weighting. Now, in practice, there isn't any clear general guidance that I can provide on choosing what the weight should be, other than saying, well, it depends on which one of the two components is more important, or if you consider them equally important, then do, as we've done in our illustration, weight.
Thomas, I think you are muted.
Yes, sorry. I was finished with my response. Sorry. Did I cut out before I finished?
Okay. Thank you.
Basically, as I said, I would take a very practical stance in thinking about, okay, which one of the PK parameters is more important, and if there is a reason to believe that one of them is more important, you would obviously include a larger weight for this particular parameter. But in our illustration, we've deemed them equally important and consequently used weights of a half.
Okay. Thank you for your response.
Okay, thank you, Adriaan. Fitene, I give the floor to you.
Okay, thank you, Helena. Yeah, thank you, Professor Thomas Yaki, for this interesting and nice presentation in this new and growing area of optimal design, especially for sparse sampling. I have a few questions. Maybe my first question is related to the previous question about the weight, the optimality criteria that we see on the screen. I was wondering whether we could also define the simple optimality criteria that you have at the top for both parameters, so that we can also incorporate the covariance between AUC and Cmax. Have you tried that approach, or?
It's a very, very interesting question. So in short, no, we haven't tried it, but I think it would be perfectly possible to define a different objective function that both incorporates variability of the parameters as well as the covariance between them. And maybe that's the point where a link to a more traditional design criterion such as the optimality could come in. One could consider, rather than the information matrix of the model parameter, one could consider variance-covariance matrix of the PK parameters and optimize determinant or whatever of that matrix in the same way as one does with DNA optimality.
Yeah. Okay. Yeah. Thank you. Yeah. My second question is about the scheme that you have, and there is a ranking, and based on this ranking, the robust criteria will give you a scheme that ranks at the top. So my question is there a big difference between the top ranking schemes or designs? Does it matter whether I will consider the optimal one, the top ranking design, or the second or the third ranking design?
So the differences between the best ones, at least in the situations that we've considered, were relatively small. So the difference between the best and the second best and the third best were moderately small. The difference between the first and the 50th, or the first and the thousandth, however, became notable. And so I can't remember from the top of my head now where in the ranking the proposed design of Chapman came, but I seem to remember it was somewhere in the region of 50 to rank of 50 to 100. So it's not a terrible design, as I said before, but if you compare the best performing to a design that's in the region of 50 to 60, you gain a couple of percent, 3 to 5% in terms of efficiency, in terms of reduction in variability, which I would claim is notable and important. But yeah, the difference between the first and the second is marginal.
Okay, thank you. Yeah, Elina, do I have time for more questions, or?
Sure, Fitena, you can go.
Yeah, okay, then thank you. Yeah, my question is about the parameters for your model. The one compartmental first order model is assumed, and you mentioned that the values of F and D are fixed. Is there any suggestion about the fixing these values, and also is it important that you get them right?
So basically what we have done is we've simply taken this model from an existing publication, just in order to illustrate how the optimality procedure works. And so D is the dose, and then and hence that has a fairly natural interpretation. And I think F, from memory now, it was defined as 1 in the actual publication. We've just stuck with that. If you think there is uncertainty about what any of these parameters are, the way to account for that would simply be to include that as one of the additional potential scenarios that you might be interested in. But as I said, this is simply an illustration that we've chosen from an existing publication, and obviously for the context that you might want to apply the optimality criterion, it may look completely different, and there may be uncertainty about what F is. And so if F is uncertain, you might want to consider different scenarios for different values of F. Similarly, you can see we have an additive error model here, and one could consider a multiplicative error model as well. This would simply be an additional scenario to consider in our robust procedure.
Okay, yeah, thank you very much.
Okay, thank you, Feteni. Thomas, in the meanwhile, I have received a question from Philip Jarvis, and he's asking whether design presented is optimized by assuming, so the design presented is optimized by assuming that information must be collected during different intervals. Is any account taken of the correlation between results for a given animal? For instance, if in one animal all the concentrations were on the low side?
Yes, so implicitly this is accounted for because we simulate data reflecting that correlation. So in our model that I have on the screen here, essentially that comes in through the error terms and the random effects of VKA and KE. So we simulate data that have the structure that we would observe in reality, and so in reality there may be, well, an animal for which all the concentrations are on the low side, and in some of the simulations this will be observed as well and hence taken into account as well.
Okay, thank you. Are there any more questions from the participants? If not, then I would really like to thank you again, Thomas, from the bottom of my heart for such a very, very interesting presentation. To everyone, I hope you enjoyed this webinar. And of course, I can only say now, Merry Christmas, Happy New Year. And in the new year, please be on the lookout for our next webinar, which will take place on the 20th of February. Thank you.
Thank you.
Thank you, Elena.